Hello, I'm Flip Nicklin. Welcome to Humpback Chronicles. Until 1998, most of the work I did with Jim Darling was focused on singing humpback whales and behavior of singers and animals around them. And then Meagan Jones joined the group. Meagan Jones, now Dr. Meagan Jones, Executive Director of Whale Trust, joined us in 1998, and we were studying breeding behavior of humpback whales, but mostly looking at the males. Meagan changed all that. Future episodes, you'll hear a lot about how important Meagan has been in the founding of Whale Trust. But for this week, just a little about some of the work she does.
When I first joined Jim and Flip as part of the research team back in 1998, we had just learned that whales that joined singers were males, not females. This finding surprised many of us, both researchers and whale watchers alike, and led me to begin to question what role the female played on the breeding grounds. If male singers were attracting other males during the breeding season, where was the female? So as part of my PhD, I set out to better understand this mystery of the female humpback. Around the same time, I remember Flip remarking on how he and Jason were repeatedly encountering pairs of whales that were just hanging motionless underwater. Pairs of whales are a common social group on both feeding and breeding grounds, and we knew that DNA tests had revealed that these adult pairs are usually either male-male or male-female, but little was known about their social function or behavior on the breeding grounds. Were these motionless pairs male-female pairs? How did they fit into what was happening here in Hawaii? At first glance, one of the most defining characteristics about pairs is the lack of activity you see from them. Because of this propensity to rest for long periods of time, these pairs were named breath holders back in the early 80s, and the name has stuck ever since. A typical breath holding pair will stay down for upwards of 20 to 25 minutes, and we have had some stay down at least 40 minutes between breaths. After they dive, they usually stay in one place and don't return back to the surface for at least another 20 to 40 minutes, and this prolonged resting behavior can repeat for hours on end, at least until another male or multiple males wander into their territory. So you can probably understand why we know so little about them. No one, researchers or whale watchers, had stayed around long enough to figure out what they were doing. So my job was to try to figure this out. And this became part of a story in National Geographic magazine in 2008 about Whale Trust work with humpbacks here in Hawaii entitled, "What Are They Doing Down There?" What we know about females is that they come to Hawaii for at least two reasons. One is to maximize their mating opportunities so they can return to the feeding grounds and prepare for birth the next year. And the other, of course, is to give birth and/or raise their calves. The primary objective of females with calves then is to ensure calf development and survival, whereas females without calves are primarily concerned with successful conception and balancing that need to conceive with the highest quality male while spending the least amount of time away from areas with access to food. What we didn't know was whether these seemingly different reproductive objectives would lead to differences in how a female responds to and interacts with a male on the breeding grounds. Did female reproductive status, that is, the presence or absence of a calf, influence how males and females interacted? We predicted that it would. We predicted that female humpbacks would behave and interact with males differently based upon their reproductive status or the presence or absence of a calf. So to test this hypothesis, we tracked and followed females both with and without a calf to see what a day in the life of a female humpback was like. We tracked mothers and calves, both with and without an escort, and females within male-female pairs. We recorded their movements, their behaviors, their social interactions, measured their body size and that of their male companions. We took DNA samples to confirm gender when a calf was not present, and we played back sounds produced by males in multiple male, surface active or competitive groups to see whether or not females responded differently to males based upon their reproductive status. By sexing both individuals within these breath-holding pairs, we were able to determine that these motionless breath-holding pairs were indeed male-female pairs, and they were much more interesting than their name implied. While male-female pairs and mother-calf escort groups can appear similar, at least from the surface, there were some subtle and some not-so-subtle differences that gave us clues as to the importance of these pairs within the mating system. Male-female pairs were typically sighted earlier in the season, and at least the females within these pairs stayed for shorter residency periods in Hawaii, allowing them greater access to food resources in higher latitudes that are critical for successful reproduction to occur. Females in pairs also spent significantly more time resting and less time traveling than we found with females with calves. And we also know that this enhanced period of time resting has also been associated with periods of estrus in other terrestrial mammals. Breathholding pairs were often the starting and ending point of a competitive or surface active group where males are known to compete for access to a presumably estrus female. In fact, at least in our study, females in pairs were twice as likely as mother-calf groups to be found in multiple male competitive groups. Now, in both male-female pairs and mother-calf escort groups, we found that males would vigorously defend their female companion regardless of female reproductive status. But we also found that males within these pairs would often appear overly alert, including coming up to breathe more frequently than their female companion. In these situations, the males would come up several minutes before the female, completing a half or full circle at the surface while the female rested below. And after completing that circle, the male would dive back down to her, and then shortly thereafter, both would surface together several minutes later. In other mammals, circling behavior and shorter downtimes in males has been associated with mate guarding, where males will closely attend a female during periods associated with high fertility and prevent potential rivals from interacting with the female. When we conducted the playbacks of male social sounds to these pairs, we observed males doing the same circling behavior around the female underwater, something we never saw with males escorting mothers. This male behavior is suggestive of mate guarding and certainly speaks to the alertness of the male to other males in its nearby surroundings. While the males became antsy during these playback trials, the females within these pairs continued to rest, and in the majority of cases remained motionless. The opposite reaction was found in female calf groups, where mothers responded faster and swam further and faster away in avoidance of these male sounds. The differences were striking. None of the females identified in our study were sighted with and without a calf in the same season, suggesting that these male-female adult pairs are primarily reflective of reproductive unions and are an integral part of the mating system in humpbacks. To support this, we now have several examples in our catalog of females observed in a male-female pair and then resighted the following year with a calf. While all of these observations suggest that these pairs represent a form of mate guarding, what we still don't know is whether the male is guarding the female before, during, or post-conception. So in sum, we found that as expected, female reproductive class did not affect the prevalence of associations between males and females. In other words, males accompanied females regardless of their reproductive status, but it did affect and impact how females interacted and responded to males. The most critical gap and limiting factor in these and all studies of reproductive behavior in humpbacks is our current lack of knowledge around the female estrus cycle. We are currently working with Dr. Nick Keller at Southwest Fisheries Science Center to help reveal how reproductive hormones may be influencing these behavior and interactions and how they may influence some of the variability we see between groups. We can't wait to share those findings with you and hope that will be sometime soon.
When Meagan joined us in 1998, a good part of our support was coming from National Geographic's Grosvenor Council. And especially, I'd like to thank Pat Cochran and Karen Kunzelman, who were supportive then and have continued to be part of Whale Trust as it developed, and they are still on our board of directors. Thank you all, and thanks for your support.